Good afternoon. I'm Dr. Brian Shapiro, a scientific writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. It's entitled Enable Your Drug Development with cGMP Cell Banking and cGMP Storage, presented by Dr. Shalmika Jackson. Dr. Jackson is a marketing segment manager at ATCC. In this presentation, Dr. Jackson will discuss ATCC's new cGMP services, highlighting how you can avoid time and labor-intensive in-house production and eliminate associated risks and costs. If you have any questions for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Dr. Jackson.
Thanks, Brian, and thank you to everyone who has taken time out of your day to attend this webinar. Just a few points to mention about our company. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and GMP services center in Gaithersburg, Maryland. For nearly a century, ATCC has been the trusted global source for cell authentication and uncompromised quality. We set the benchmark for providing cells used to discover and develop some of the most notable advancements in life science. Now, we are pleased to announce that we can provide these services under current good manufacturing practices, which is a natural complement to our well-established GMP biorepository storage services. Today's talk will begin with a bit of a history lesson in that I'll describe how GMP originated out of a dire need to protect consumers and regulate the manufacture of food and drugs in the U.S. Then I will define what GMP is and why it's important. I'll briefly touch on some of the prominent GMP regulations and how they impact drug manufacturing, including small pharmaceuticals and large molecules like biologics. I also want to spend some time diving into the manufacturing of biologics and the importance of cell banking. Then I will finish the webinar with an introduction of ATCC's new cGMP cell banking services and how coupling cell banking with our cGMP biorepository can help to preserve the security and integrity of critical starting materials during the life cycle of drug development and commercialization campaigns. Around the turn of the last century, a chemist at the United States Department of Agriculture, Dr. Harvey Wiley, in his crusade for safer products, established the Poison Squad, which was comprised of a group of volunteers that agreed to eat only foods treated with measured amounts of chemical preservatives like borax, formaldehyde, and salicylic, sulfuric, and benzoic acids. Essentially, they performed one of the first highly controlled in-human clinical trial studies, which eventually became a national sensation and raised awareness regarding the need for food safety. Throughout his career, Dr. Wiley waged continuous warfare on adulterated foods and drugs, and he ultimately became the first commissioner of the United States Food and Drug Administration. Now, a few years later, in 1906, Upton Sinclair, who is the epitome of a muckraker, which in today's language we would call a watchdog journalist, published The Jungle, which entailed a graphic exposure of the meatpacking industry. Public outcry resulting from the filthy conditions described in his book, coupled with Dr. Wiley's unceasing advocacy for national pure food and drug legislation, pushed Congress to pass the Pure Food and Drug Act of 1906. Now, while this law was a great step forward, it nonetheless left gaps in consumer protection in that it did not allow for proactive compliance requirements. Labels were not required to state weight or concentration. It was only required that a contents statement, if used, be truthful. Even after a slew of false therapeutic claims, Congress refused to take action until a tragedy occurred that took the lives of over 100 people, mostly children. In 1937, an elixir of sulfanilamide, a drug used to treat streptococcal infections, was marketed to children with sore throats. The company tested it for flavor, appearance, and fragrance and found it acceptable. However, they did not anticipate that many people who took the drug would die of terrible deaths associated with kidney failure, urogenital and gastrointestinal malfunction, and convulsions. The chemists had used diethylene glycol, which is a primary component of antifreeze, to dissolve the solid sulfanilamide into a liquid. At the time, there were no requirements that drugs be tested for safety. This tragedy propelled Congress to overhaul the 1906 law, which had laid the foundation of the Federal Food, Drug, and Cosmetic Act of 1938. It required that drug manufacturers show that a drug is safe before marketing it. In addition to drugs, cosmetics and medical devices were regulated for the very first time. The FD&C Act regulated all poisonous substances and granted the FDA the authority to inspect factories. Now, it took 20 more years for the government to require that scientific evidence be used to support drug safety and efficacy and claims made in advertising and labeling. Enter thalidomide. Despite disturbing findings, Congress once again did not pass legislation until a near tragedy struck. This time, tragedy was avoided due to the diligence of a woman pharmacologist working for the FDA, whose name was Dr. Frances Oldham Kelsey. While she was a university faculty member, she observed that some drugs pass through the placenta during pregnancy. One of her first assignments at the FDA was to review an application for thalidomide, which was marketed as a tranquilizer and a painkiller, but was also used in pregnant women for morning sickness. Even though thalidomide was approved in many countries, Kelsey withheld approval and requested additional studies because she was concerned about the drug's impact on the nervous system. Around the same time, many babies were born with severe deformities in Europe and other places. When these deformities were eventually traced to the teratogenic effects stemming from the use of thalidomide during pregnancy, Kelsey became a hero for keeping thalidomide off the U.S. market. Because of the high-profile nature of the thalidomide incident, public opinion pushed Congress to unanimously pass the Drug Amendments of 1962. These amendments tightened control over prescription drugs, new drugs, and investigational drugs. For the first time, efficacy or effectiveness now had to be shown before a drug would be approved. It was the Drug Amendments of 1962 that formalized good manufacturing practices that we use today. So what is GMP? To put it plainly, GMP is a collection of government-enforced requirements for methods, facilities, and controls used in manufacturing of drugs, which also extends into the final processing and packaging of drug products. GMP ensures that products meet rigorous specifications and quality and regulatory standards. Quality is built into each step of the process, and documentation of each step is the evidence of that quality. GMP specifically covers requirements for QC in production, which is a matter of paramount importance to protect the end users, the patients. So why is GMP important? From a humanitarian perspective, GMP ensures public and consumer safety. In terms of risk mitigation, adherence to GMP procedures aids in minimizing potential risks, both known and unknown, that could have deleterious effects on human health, as was often observed prior to the drug amendments of 1962. Taken together, cGMP is a necessary component to ensure safety, efficacy, and quality of life-saving therapeutics in efforts to reduce overall health hazards. And from an economic perspective, for businesses, health insurance is the largest component of non-wage compensation. And for employees, healthcare is one of the largest categories of consumer spending. So, by demanding that drugs are manufactured under GMP, consumers gain a sense of assurance that their hard earned money is not wasted on products that have little or no medical value. Now we will move on to some of the key GMP regulations for drug development. Title 21 is the portion of the Code of Federal Regulations that governs food and drugs within the United States for the Food and Drug Administration. Now that we're in the digital age, Part 11 covers the implementation of electronic records and signatures. It provides guidance for validation, audit trails, record retention, record copying, and legacy systems. Parts 210 and 211 address the manufacture of pharmaceuticals and requires that all medicinal products are fit for their intended use in compliance with the requirements of the marketing authorization and do not place patients at risk due to inadequate safety. 600 and 610 cover biological products such as monoclonal antibodies, vaccines, blood, and blood-based products, and the standards that are used to ensure safety and efficacy. Part 820 covers medical devices from simple devices like syringes and catheters to implantable devices like pacemakers and even software that's used in computer-aided diagnostics. Taken together, GMP ensures that people comprehend their roles and responsibilities. Products have clear specifications at each phase of production. Processes are properly documented, simple, and consistent. Procedures are in place to establish guidelines for understanding and implementation of critical processes. All of these are underpinned by the manufacturing premises, which should always maintain a high standard of cleanliness with validated and calibrated equipment to ensure a safe and effective drug product. Let's dive into the manufacture of two types of drug modalities, small molecule drugs like aspirin versus large molecule biologics like monoclonal antibodies. Now, there's a common misconception that manufacturing a medicine can be relatively straightforward. You initiate a series of automated steps and the product comes out. In reality, the process is incredibly complex. Let's start with aspirin, which is the most widely sold over-the-counter drug. It's a relatively small molecule, having a mass of only 108 Daltons. In contrast, a typical monoclonal antibody has 1,000 times more mass, topping the scales at 150 kD. Think of a regular bicycle that weighs 20 pounds and has roughly 150 parts. In contrast, an IgG antibody would be analogous to a private jet weighing in at over 25,000 pounds and is comprised of 6 million parts. The production of aspirin is relatively straightforward. It involves the reaction of salicylic acid and acetic anhydride, which is crystallized to produce aspirin. The drug substance goes through a series of filtration steps to remove the acetic acid and solvent. And then the crystal aspirin is then sifted, granulated, and tableted, and then voila, an effective treatment for pain, fever, and inflammation since 1953. Now, if there are any card-carrying synthetic chemists attending the webinar, please excuse any oversimplification. However, biologics such as therapeutic antibodies, which are made in living cells, are thousands of times larger and more complex than relatively simple molecules like aspirin. The complexity of biologics inherently affects the complexity of the manufacturing process. In addition, the fact that biologics are produced in living cells poses a number of interesting challenges. For example, no two living systems are exactly the same, as they're prone to natural variation, which is the primary reason biologics manufacturers must start with master and working cell banks produced by highly trained personnel, along with appropriate custody documentation to ensure that their cells are maintained under controlled conditions from the inception of the project. Up to half of a regulatory application for a new biologic focused on the manufacturing process. That's because regulatory authorities like the FDA or EMA are not just licensing the product, but the exact way in which it is manufactured. The first step in manufacturing biologics is genetic engineering. Introduction of a sequence of DNA into the cell, which acts as an instruction manual for the cells to create the intended biologic. This single cell is expanded to generate thousands of identical copies called a cell line. A single vial of a particular cell line is thawed and allowed to grow in a controlled environment until the cell population grows into the trillions. In fact, in a single 12,000-liter bioreactor, there are more than 100 trillion cells working side by side, acting as tiny factories to produce a biologic. This number of cells is more than 1,000 times as many stars found in our galaxy, the Milky Way. Cells are then grown to sufficient numbers and then transferred to large-scale production tanks until it's time to harvest and purify the biologic. The biologic drug needs to be separated from the cells, any unwanted proteins, and other process-related impurities. And then the purification is also a multi-step process, and it varies from one biologic to another, unlike simple chemical synthesis, whereby a single process can be used to make many different derivatives. As you can see, biologics manufacturing is riddled with challenges, but holds so much promise for revolutionizing treatments of several serious illnesses. And this promise is being realized by overcoming the scientific, operational, and quality and regulatory hurdles. As mentioned before, the inherent biological variability of cell culture bioprocessing, coupled with the physicochemical characterization of biologics, are the primary scientific challenges. In terms of operational constraints, raw material qualification, maintaining a clean, hygienic area, and controlling environmental conditions are key to maintaining a robust manufacturing process. Now, quality must be baked in at the inception of manufacturing a biologic, following QBD principles from the very beginning. Having a robust QMS with established, validated processes is also integral to maintaining consistent product quality. Another key challenge associated with manufacturing biologics is not only understanding, but also accurately interpreting regulatory guidelines to aid in minimizing unforeseen risks. Now, biopharma companies that plan for success understand the importance of cell banking. To aid in this understanding, the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, or the ICH, makes recommendations on best practices that should be used to ensure our drugs are safe, effective, and of high quality. In ICHQ-5, there are several recommendations about cell lines that are used to produce and test the efficacy and safety of biologics therapeutics in general. It explains the importance of documenting cell line history, production processes, and testing methods. Banking the cells reduces the cost of continuous cell culture processes, which includes consumables such as media and supplements, and the time associated with maintaining cells in culture. It is just more cost effective to suspend the growth of the cells until they are needed. Now, the most beneficial attribute of cell banking is that it reduces the frequency of genetic drift in culture. We are all aware that as cells divide and proliferate, there's an increased chance that the cell's DNA could be damaged, resulting in a permanent mutation. And if that mutation is passed on to daughter cells, it could result in altered phenotypes and growth patterns. In terms of drug development, cell banking is critical in forming a solid foundation for building a biologic to ensure that the material is free from contaminants and safe for product manufacturing. And of course, a two-tiered cell bank system going from a master cell bank to a working cell bank provides a common starting source for each manufactured lot of drug product. Now, it is no surprise that the global cell banking market is growing rapidly, but the market itself is challenged by the current regulatory climate and high costs. So, what is driving this rapid expansion? Primarily, the increased expenditures on healthcare, specifically the tenfold higher price point for biologics produced from living cells compared to small molecule drugs like the aforementioned aspirin. Also, operational improvements and the emergence of outsourcing organizations that can develop, bank, and store master and working cell banks that are used to produce biologics. Conversely, the market is being restrained by increased global regulatory stringency and scrutiny of manufacturing processes and cell line characterization and testing. In addition, the high costs associated with establishing a robust cell banking and storage service are also major restraints. As innovative, novel biologics transform the biopharmaceutical market, improved processes and technological developments are likewise changing the future of manufacturing. This is the primary reason ATCC is viewed as the partner of choice. ATCC is regarded as a pioneer in cell banking and storage, and our longevity and reputation show our commitment to preserving the integrity of critical starting materials. Arguably, we are the trusted global source for cell authentication and uncompromised quality. Companies that partner with ATCC to outsource the manufacture of critical raw materials like production cell banks can utilize their resources for higher value development and manufacture of final products, thus avoiding time and labor intensive in house production and elimination of associated risks and costs. ATCC is committed to provide quality products and services to the life science community. This commitment includes the following certifications and accreditations: ISO 9001, 13485, 17025, and 17034. Our experienced quality management team ensures that the acquisition, authentication, preservation, development, and distribution of biological materials are performed under and conform to a documented quality management system. We adhere to the regulatory requirements through the use of robust quality control testing, exceptional documentation and record keeping practices, corrective and preventative actions, internal and external audits, and management reviews. At ATCC, we are transforming to serve an evolving life science community. In 2021, we commissioned a new purpose-built facility in Gaithersburg, Maryland, hold the distinction of being 21 CFR 600-610 to produce master and working cell banks. ATCC is widely known as the home of cells. Our GMP services team solely focuses on mammalian cells and stem cells. With over 5,000 fully authenticated cell lines, GMP customers have access to the most expansive collection of authenticated cell lines in the world. Or we can scale up proprietary cell lines to produce banks that ensure process robustness over many manufacturing or testing cycles. Also, customers can safely store their cell banks in our cGMP biorepository during the life cycle of their project. Our capabilities enable us to support many customers within biopharma, such as biologics manufacturers, customers developing cell-based assays to assess the potency of their drugs to determine efficacy, and customers developing in vitro toxicology assays to ascertain the safety profiles of their drugs. In addition, cell and tissue manufacturers that need to expand or store their human cells, tissues, or tissue-based products that are used for implantation and transplantation, infusion, or transfer studies. Our cGMP process begins with customers either sending us a few vials of their research cell bank prepared from a selected clone under defined conditions, or customers can select from a menu of over 5,000 fully authenticated cell lines from a variety of tissues, diverse range of disease states, and molecular signatures. Typically, a single vial will be selected to create 200 to 400 vials of a master cell bank. A master cell bank is a single pool of cells that has been prepared from a selected clone under defined conditions. The master cell bank is used to derive all working cell banks. The working cell bank is prepared from aliquots of a homogeneous suspension of cells obtained from culturing the master cell bank. Customers also have the option to have a working cell bank of 200 to 1,000 vials created under GMP. Also, customers can safely store their cell banks in our cGMP biorepository for the entire life cycle of their campaign. Our cGMP biorepository experts can characterize, package, store, and distribute biological materials to ensure the security and integrity of cell banks. With a variety of temperature options available, we have the capacity to not only store cell lines, but also microbes, proteins, and nucleic acids too. Customers around the world have trusted us for nearly 100 years to safely segregate myriad biological materials. GMP regulations grew out of legislation passed because terrible tragedies either killed many people or almost did. GMP ensures that medical products we use are safe, pure, and effective. Biologics are one of the fastest growing classes of therapeutic compounds, and they promise targeted treatments for a range of conditions. However, biologics manufacturing is complex and riddled not only with scientific, but also operational, quality, and regulatory challenges. In terms of drug development, cell banking is critical in forming a solid foundation for building a biologic to ensure that material is free from contaminants and safe for product manufacturing. It is important to use authenticated cell lines with established pedigrees under validated cGMP cell banking procedures. And by depositing your cell banks with ATCC, you're choosing the global leader in long-term biomaterial storage and management. For more information about our cGMP services, please visit atcc.org/cGMP. And at this time, I will gladly take any questions.